만나고 헤어지는 모든 일들이 어쩌면 어린애들 놀이 같아. 슬픈 동화 속에 구름 타고 멀리 나르는 작은 요정들의 슬픈 이야기처럼, 그러나 우리들 알지도 못하고 울지만 사랑은 아름다운 꿈결처럼 고운 그대 손을 잡고 밤하늘을 날아서.
Hi everyone, how have you been? So, in continuation of the chapter 5, which is about the DNA actions. So, last week I have just briefly introduced, I was about to actually introduce this DNA profiling and actually the mentioning of this heterochromatin area, which I mean the constitutive heterochromatin, that constitutive heterochromatin usually the two most outstanding area of any eukaryotic chromosomes is actually this. If this was a chromosome, then this telomeric area, the ends of chromosome, telomere, and the centromere, the center. So around about this centerish area, we usually designate and call it a centromere. And these two regions are the one usually considered to be this constitutive heterochromatin area, which the constitutive means that is always 24/7, no matter what happens. These areas are heterochromatin means those structure of a chromosome is highly condensed. In terms of we have already looked at the this chromosome organization mediated by this nucleosome, so that every DNA double helix in a eukaryotic gene is actually the unit of this nucleosome. Basically, the protein, the histone protein, 4 different kinds of histone proteins make up this nucleosome core structure and then wrap DNA around which, and this nucleosome unit, by just like accordion, you can just expand and then shrink depending on your need. So when those chromosomes are tightly wrapped around, so that highly compact, when they exist in a highly compact structure, we call that as an heterochromatin. Whereas when it is also possible the same region, this nucleosome, actually the way they can play this, the magic is have a relative distancing among these different nucleosome unit, and then you can have a relatively relaxed, uncondensed chromosome structure. Then we call that as a euchromatin. So actually this type of a chromosome structure is a typical chromosome structure that we know of, is highly condensed. So actually this chromosome is perhaps the most the highest condensed structure of all among the range of different possibility of this chromosome condensations. But even what we say is, even in the status of chromatin. By definition, our genome can, in its structure, they can switch between chromatin, which is a relatively relaxed structure, and the chromosome, which is a highly condensed structure. So the point is technically, to say more accurately, the chromosome can only be observed just right before cell division. So only when a single cell is about to divide, split into two, that means your genome must be packaged and then split into two equal halves. Then is only when can you see your genome in this chromosome structure. But during the rest of the period of your cell lifespan, your DNA exists in this relatively uncondensed structure we call as a chromatin. However, on this chromatin there are two different degree of condensation. One is relatively highly condensed, then we call it as a heterochromatin, and the other one this is eukaryote, heterochromatin. So then this heterochromatin is highly condensed, and on the other hand, when it is more relaxed, loosely organized, then it is a eukaryochromatin. So speaking of this heterochromatin, constitutive means always. So it doesn't really change its structure, although it can theoretically. Then they are constitutive heterochromatin area, and they are at the end of any given chromosome. Eukaryotic chromosomes are all linear, mind you, and the center area is a centromere. Is the point. Why do I bring up that heterochromatin thing? And those, another thing is, those heterochromatin area is usually they don't have any genes. Later today, we are going to take a look at how do we actually define a gene. What is a gene and what is non-gene, technically speaking. But before we just do that, these heterochromatin areas, especially those are constitutive heterochromatin area, they don't harbor any gene. I mean, if at all, there are only a few genes, not too many genes, and there is a really actually specific correlation. There is a reason for that. Why you don't have any genes in the heterochromatin area. Later we will try to take a look at and try to understand why there is such a link between chromosome condensation and the genes. And also, no gene, you don't have any genes, and because of that, actually, that's the point that last week's lecture, because of that, the DNA sequence variation among different individuals are very high in this heterochromatin area. Why is that so? Why those specific area on the DNA, why, if that area does not have any genes, then your DNA sequence can vary so much, whereas those areas occupied by the genes, you don't really actually have too much degree of freedom to be different. Why? Because those areas occupied by the genes, if you have any sequence variation, which means any sequence variation, or each single sequence variation potentially the mutation. Yeah. And the source, the reason why you have a sequence variation, why your DNA sequence in certain specific spot is not identical to the other guy. What happened? As a result of the mutation, right? And most mutation are bad. Like usually the chances are if the genes, if those DNA was serving as the area as genes, that means you have some function, right? Usually, like for example, you're making some very important protein, for example, hemoglobin, right? Hemoglobin is a protein which is extremely important for all this animal, like blood, having this blood circulating animals. For them, hemoglobin is extremely important because hemoglobin's function is to carry oxygens to every different part of your body. Yes. And then there is a gene actually making this protein, so that's the hemoglobin gene. And if you have a DNA sequence variation, then chances are most likely then you would actually screw the fine structure of your hemoglobin protein, and usually if you alter the structure, then the function will be affected in negative ways usually, right? Only very rare occasions those such alteration could somehow generate more beneficial, more functionally superior ones, and that's the very slow but meaningful step towards the evolution that has been that way. So that is the reason why in mostly sequence variation in the area of the DNA occupied heavily by the genes, then if you have those sequence variation, then the carrier, the individual carrying those sequence variation will not been able to survive, or even in some cases, even if they can, but probably that they wouldn't be able to produce more number of offspring. So that's the reason why you don't really get to find any such DNA sequence variation. However, on the other hand, those areas not occupied by the genes, then you have such a ultimate freedom of it. You can be, you can have any consequences, bad consequences, without worrying about such things. Then you can have so many different variations. So, bottom line is, those area, those heterochromatin area, which means what I mean by that is a constitutive heterochromatin areas, DNA sequence variation is very high. So in other words, like human, if you compare your DNA sequence with that of Neanderthals, they have like more than 99% identity, DNA sequence identity. In other words, if you somehow pick up two individual, human individual, any given day, and then you compare their DNA sequence, then chances are they would like almost 100% identical. Then what is your variation? I mean, obviously, we all look different. We all behave different. There's got to be some kind of a DNA sequence variation between two different individuals. So mostly those variations are usually, although I'm not saying that all those gene areas are 100% identical among all different individuals. No, it is not. But relatively speaking, if you compare those DNA sequence variations, much higher in those non-gene occupied heterochromatin area is the point. And then now, moving into this DNA profiling issue, which is known as DNA fingerprinting, DNA testing, or DNA typing. Just like based upon this idea, theory that all human fingerprints represent some very unique identity, so that we can use this individuals' different fingerprint pattern in identifying different unique individual. So is the DNA sequence on those heterochromatin area is the idea. And why we can use such areas DNA sequence as if they represent some kind of a molecular DNA version of a fingerprint, as I have just explained, right? So this DNA profiling is it's been well known forensic technique identifying the specific person's DNA character. In this country, even very recently, we have such issues that like the closer we have this genetically, the closer two individuals they are, then DNA sequence or like if you compare such area of DNA sequence using this technology of DNA fingerprint, then it will match 100%, almost 100%, right? I mean, in the case of a identical twin, it's got to be 100%. If you compare this DNA sequence in terms of this DNA fingerprinting technique between mother and a child, it will be extremely similar, right? Just like that. And sibling also very similar. But if you compare such DNA area sequence using fingerprinting between two genetically unrelated individual, then the pattern will be very distantly related. I mean, it's not so much identical as those genetically related individuals. So actual here in DNA profiling technology that I'm going to right now show some of the little different technical aspects, so you may some of you at least maybe a little bit lost along with this explanation. But remember this, there are three or two keywords involving this technology in explanation of such techniques. Here is the sequence variation, of course, is the key, and how you identify those sequence variation in this specific DNA fingerprinting technique. Some of you may have a little bit of a wrong idea about we are actually do this DNA sequencing. DNA sequencing means like let's say certain region of DNA, you have A, G, A, C, C, A, C, T, A, G, like this DNA, and then you identify each nucleotide, what type of nucleotide you have on a particular region is the sequencing. Actually, the human so-called the famous human genome project, which was done in the year, which was actually completed in the year 2000. It took 10 years at the time to complete. On the human genome project, there was something that one by one, what type of DNA sequence we have as a human. So 3.3 billion DNA nucleotide base pairs, entire 3 billion, more than 3 billion nucleotide bases were, for the first time, were read and then identified what type of the order in terms of DNA sequencing. That was the DNA sequencing. Yeah. Maybe if we had a time, maybe we could look at the theoretical and technical basis of this under this DNA sequence technology. But I don't really think we have enough time, and it's even necessary. So it's a suffice to understand the difference between DNA sequencing and this DNA profiling, which does not even involve actual DNA sequencing. No. So on the DNA profiling, the keyword is you are actually taking advantage of some of the repeat. Actually, this repeat means DNA sequence repeat. To be more accurate, it's called tandem repeat. So this is one keyword that you might want to take notice. DNA profiling, you are trying to identify what type of DNA repeat pattern one individual has. You identify in terms of the repeat number. How many repeats you have? What I mean by the repeats, let's say in a certain spot here, certain area of your DNA. Let's say I will just, this is one region, and this region, this block consists of some of DNA sequence, AGG, AC, for example. This is to give you an example. The same AGG, AC, this sequence, mysteriously on this particular region, not once, but not twice, but several times, the same DNA sequence repeat appears, like as if you have some of the block module, just like this. This, what I mean by this tandem repeats. And the interesting thing about this is this repeat, the number of repeats. Here 1, 2, 3, 4, 5, 6. Here repeat number is a 6. Number of repeat varies in different individuals. So if a person A on this particular area of the DNA in this region, this particular DNA sequence, AGGAC, nucleotide sequence repeat, if this person has a 6 or the exactly same location of the gene, another person has maybe he or she may have a 7 or 9. So the point is, everybody has different repeats. Or the thing is, maybe another person have the exactly same number of repeats, 6 over here. However, this type of repeat sequence, so everywhere on your genome, like how many chromosomes you have a 23 chromosome, and the diploid number of your chromosome is a 46, and sometimes even between two homologous chromosomes, this number of the repeat could be different. So in all of combination, there is no way one person has exactly identical number of repeat altogether with another person. That's the key. Unless these two individuals are identical twin, even this number of repeats is not going to be identical between a mother and a child, or the father and child. So by determining how many number of repeats you have in particular region, and then you have whole collection of this test among all 23 different chromosome, haploid number chromosome, and also you compare that another half. So all pattern of these repeats into the homologous chromosome in your cell, then you can generate only in terms of these numbers, you can generate a very unique piece of information which is tightly linked to your personal identity. That's the key point of this DNA fingerprinting. So here, in order to understand and in summary, to understand this DNA profiling technique, the first keyword that you might want to carry is this tandem repeat, okay, which is not equal in different individual. The second key point is here, how do you identify how many repeats you have? Obviously, you cannot count each repeats that so easily. If you stop for a moment and try to think of how do you know actually how many repeats you have in any given individual. Here comes the concept of the probe, which is actually not only in this DNA profiling but in many different very important molecular techniques, including this PCR technology, which has been the basis for this coronavirus testing these days. All this concept of this probe has been instrumental in, but actually this concept of the probe is not really that like a rocket scientist, too complex technology. It is extremely simple concept, but that simple concept dictates so many different areas of this molecular techniques, and that has been the driving force of the whole this progress in this DNA technology. So having said that, let's get into this. So this so-called the combination of alleles we have already looked at what's the alleles. Alleles means some of the different version of a certain DNA sequence on your area, the locus. Okay, if we consider this tandem repeat area as one of the such locus, because any location in the, although usually classically the locus has been used in designating the location of the genes on your chromosome, and the repeat regions, they do not play as any genes. They are not at all. They are not genes. However, still we could not come up with any other better wordings for this, so simply just include this area as also known as an allele. That's how we do. So the combination of these alleles in various tandem repeat loci of a person is raw material, so to speak, raw material for this DNA profiling. It is not something that the wording that I just made up. If you look up and reference, this is how it's been described. So what is the combination of the alleles in various tandem repeat? The one thing that we could at least try to figure and hopefully understand is this tandem repeat, because I have just explained what they are, the tandem repeats. And this tandem repeat, what do I mean this combination? All these alleles, which means even in a given chromosome, you may have this tandem repeats. Tandem repeats, one kind of tandem repeats, number 1 here. And you may also have another tandem repeats of a different sequence of a B over here. But if this is a chromosome number 1, in the case of a chromosome number 2, you may have another such tandem repeat, let's say just a C, and there is another D. So and this is the case of chromosome number 2. So it can go all the way up to chromosome number 23, which happens to be in X chromosome or Y chromosome. So maybe at least the chromosome number 22 you can go all the way up to. Yes. And so then if you combine all together, then you create some very important and also very huge variation of such data as what it is saying. So bottom line is, what you can say about this DNA profiling is unique DNA sequences. Yeah, it is actually unique DNA sequences, but it exists as some kind of a set in terms of a different repeat numbers also. So unique DNA sequences sets of present in different people. So different individual is what they are. So that displays so-called DNA sequence polymorphism. Polymorphism means it shows all differences among different individuals. Is basically what says about polymorphism, right? So because of that, now it is possible to identify a specific individual just like the fingerprint usage. So actually almost 100%, about 99.9% of DNA sequences are identical among different people. However, what matters is this very small 0.1% is the unique sequences, and still this 0.1% take up huge in its actual sheer numbers. So it is just more than enough to be able to serve as such a point or to leverage, I should say, leverage with which you can distinguish all the individuals. So that's what I'm saying. This is tandem repeats. So how do you identify these tandem repeats? But before we do that, first, how do you identify any DNA sequence variation in a given spot? Let's say, for example, once again, let me give you an example. Let's say in the chromosome, particular chromosome, there's a locus, right? One locus area. So if you zoom in this locus, zoom in, and let's say this is a gene. Okay, a gene is there in a locus. So person A and another person on the same chromosome, same area. Okay, so it's gotta be another gene, which is the same gene. However, let's say the gene consists of some DNA sequence. On this particular area, this person A, this particular spot, the DNA sequence is GAACCT. Okay. However, this person B, this DNA sequence has been mutated into GATCCT. Okay. So we are talking about just one DNA nucleotide difference. Okay. This is a kind of a basis for DNA sequence variation, right? So how do you actually, in the first place, we are not even jumping into identifying technically in practice, identifying different number of tandem repeats that I say everyone is supposed to have in different numbers of repeats. But before we get into that, let's take a look at this first. You have these two individuals having one DNA nucleotide sequence change between these two individuals. One has an A and the other one has a T. Other than that, you have almost identical GAACCT versus GATCCT. Wow. So in the technicality, the way the technique by which this change in DNA sequence was detected has been classically, traditionally, has been the principle for this whole DNA profiling. Okay? How is actually RFLP? RFLP stands for, maybe later on, probably the full name will be shown up in slide 2, but RFLP is standing for restriction F fragment, L length, P polymorphism. Right. So polymorphism, I have just briefly mentioned a minute ago. So this polymorphism is they are all the variation, high variation among different individuals. Basically means polymorphism. And the thing is here that we need to understand is a restriction fragment. What is its restriction fragment? And its length. So here, basically RFLP, what RFLP stands for is, whatever it is, that there is something called a restriction fragment. What is this? Restriction fragment, okay? So at least you know what the fragment is, right? Fragment. And this is whatever it is, it's called a restriction fragment. And their lengths, the restriction fragment lengths, are all different, which means polymorphism in different individual is RFLP is meaning. So you can detect these different lengths, the fragment lengths, and by virtue of, oh, you have this long fragment versus you, another individual, you have a shorter fragment of the same locus region is the basic idea. So it's RFLP, the another way of explaining a little more officially is a RFLP can distinguish the DNA sequence variation which was generated as a result of mutation among different individuals. How? By the different sizes of these DNA. Differences in the sizes of a DNA fragment. And what DNA fragment? Cut with this. Now you have to understand this restriction enzyme. This restriction enzyme is like this. This is restriction enzyme. What is this? What I mean by that? I mean, this is a scissor. How can I not draw any scissor? It is a molecular scissor. You can cut your DNA fragment at will on a very specific given DNA sequence. That's what this restriction enzyme does. So if you cut this DNA fragment, then this particular area will have certain lengths of DNA fragment. But the restriction enzyme, enzymes can only cut a specific region of DNA based upon its DNA sequence. In other words, a specific given restriction enzyme can only cut only if that DNA sequence has some very specific sequence motif. Then this restriction enzyme can cut this. But then, as a consequence of a mutation, if that DNA sequence has been changed into something different, then this restriction enzyme that used to be able to cut can no longer cut that area. So there is a very high DNA sequence specificity exists for all different types of restriction enzyme. That's as best as I could explain about this concept of restriction enzyme. Before we go into further, I will explain further, taking the example of this mutation in the hemoglobin. But before we do that, what about this RFLP, which stands for restriction fragment length polymorphism, right? This RFLP is actually a very famous the biological science term. So people, especially in this era of DNA or recombinant genetic engineering and molecular biology, the people within this inner circle, INSA, people casually refer this RFLP. However, they somehow call this RFLP instead of RFLP. They just RFLP. They pronounce this as a RFLP. So if you also somehow get the habit of pronouncing this RFLP as a RFLP, then you may actually look like some of the experts, the people who know something about it. But anyway, so this RFLP, to give you a little further details information about how this RFLP can be done using whatever it is, this restriction enzyme. Let's take the practical specific example, the real life example here. This is the classical example from which actually the concept of this RFLP arose for the first time. Okay. So in hemoglobin, that the important protein carrying the oxygen in our blood, those protein only present in red blood cells. The hemoglobin actually consists of two different types of protein. One is called alpha globin, and the other type of protein is a beta globin. In terms of its protein structure and the DNA sequence, they are highly similar. However, it's not identical, 100% identical. So these are technically two different genes as well as two different proteins. So alpha and beta globin, two proteins, two highly related proteins, combined together. Actually, two of each of identical alpha globin and two of each beta globin, so four proteins get together to make up one entire functional hemoglobin protein, as you see in this the structural model here. Here in blue, the ribbon-like structure. Usually, these DNA protein structures are usually being represented in this ribbon model, as I have shown you several chapters ago. So this blue, whatever they are almost very highly similar in structure. So if these two blue proteins and an alpha protein, and maybe these two red proteins, the beta protein. So anyway, two alpha and two beta globins are the ones that making up this one single functional unit of the hemoglobin. And so some individual carry mutation on the beta globin gene. A very small, one single DNA has been changed into other. However, then what happened is you have this changed DNA sequence, and you have this particular the defect and sickle cell anemia. Due to that one single nucleotide change, then the structure of the beta globin protein is changed into something drastically very different. So because of that, the original hemoglobin structure of like this globular shape, the round shape, is drastically altered into highly different one. So then hemoglobin, that mutated hemoglobin protein cannot function normally, and even the red blood cell carrying such mutant hemoglobin protein become very different in its structure. Like now its shape is something very weird, crooked form, whereas normal red blood cell is like very flat, a donut like shape. It's not entire donut with hole, but maybe flattened cushion, or whatever your view is, just totally up to you. The point is, the sickle cell red blood cell is totally different in shape, and inside of which you don't have any the functioning hemoglobin protein. That's the point. But the reason for this entire drastic change is only just a single nucleotide conversion from one to the other. Okay, so it's not really fair that with such a very minor change should have such a huge consequence. Anyway, so this change, DNA sequence change, how do you detect this DNA sequence change without having any entire DNA sequence sequencing conducted on that region? In these days of highly advanced technology in the molecular biology, maybe you could even try to attempt to sequence quickly the entire area of the hemoglobin gene, but even long time before such technical advances, scientists were able to detect easily, relatively speaking, this DNA sequence alteration, thanks to the presence of the restriction enzyme. Restriction enzymes are, like I said, those enzymes that can cut certain area of DNA specifically based upon the specific DNA sequence. So if this particular area, the DNA sequence changed, then probably the one particular restriction enzyme that was able to cut no longer able to cut that particular area. So the DNA fragment which were cut by the restriction enzyme would be different in length is the point. And that's what I meant by this RFLP, restriction fragment length polymorphism arose here. So let's take a look at this example here. In this wild type means so-called unmutated. Usually this wild type is referred as normal, but what is normal and what is abnormal? That's the reason why I really hate using the term normalcy. So rather I would like to stick with this unmutated gene here. This is the name of particular restriction enzyme that was able to, so this is the entire region of this beta globin gene, human beta globin gene, and there is a 4 location, 4 location that the target of this particular restriction enzyme DDE1 is named. That means these 4 locations have the same DNA sequence. All of which can be recognized by this same type of particular restriction enzyme DDE1. Okay, then if you apply your DNA sample, you isolate your DNA from your cell, and then expose this particular area, expose to this particular restriction enzyme. By the way, all these restriction enzymes were purified from bacteria. These bacteria are human don't have any such a this type of restriction enzyme, but bacteria have. Why? Because they kept this restriction enzyme in their defensive weapon against invading viruses. So whether you're a bacteria or human or an animal or even plants, they show, all of them show the really fierce history of struggle and battle, fight against infecting viruses. Certain viruses only infect a plant, certain viruses only infect the bacteria, and certain viruses on the other hand infect only animals, including humans, like that. So no organism can escape from this infecting viruses, such as horrible genetic elements the viruses are. Anyway, so this particular restriction enzyme can cut this 4 location on the beta globin gene, and as a consequence, how many? If you only focus on this area, okay, covered by beta globin gene, then you have 3 fragments. Like if you can somehow detect this area after the cutting by this restriction enzyme, particular restriction enzyme, you will have 3 different fragments, DNA fragment. One is about 175 base pair, another one is about 200. It's not really easy to distinguish in length, fragment length, between 200 and 175, but it is not impossible. You still can have a little bit of differences, and so you have 1 fragment and 2 fragment and another 3rd fragment, so fragment 3, and this the third fragment is the largest fragment. There's one large fragment. However, in the mutant beta globin allele, this particular site is missing, so it's gone here. So what happened is, because of that, you have only one fragment combined together, these 175 and 200 base pair combined, so it's like intermediate fragment is generated, and still you have one large fragment. So in this individual, mutant individual, you have only 2 fragments instead of 3 fragments. So this wild type, 3 fragments. And in mutant, you have 2 fragments. Because due to the absence of this coding site due to the sequence change, so this enzyme cannot cut over here, and that's the reason why you have this only 2 fragments. And obviously those fragment lengths are changed, so that's the basis of this resection fragment length polymorphism. And how do you distinguish? How do you distinguish these differences in fragment lengths? Using some technique called gel electrophoresis. It's called gel electrophoresis. Basically what it is probably I will just get to that a little more detail a minute later. But the thing is, here if you load your DNA sample, in these two, look, this is what some mesh-like the material through which your DNA can migrate through it, because this is like some soft material, soft solid material, so that's why we describe this as a gel. So this is kind of a gel, a flat gel, but this material contains small pores, and through those pores DNA can move around, and you apply this gel on electricity. So here at the bottom positive pole, and on the top negative pole. Then if you apply initially your DNA sample over here, okay, then DNA will migrate toward the bottom of this figure, because DNA and also RNA, they are all negatively charged due to the phosphate group. So this negative charged group will be attracted to the other end of this electrical field. So that's the kind of a little bit driving force for this movement of nucleic acid. But there will be some kind of a speed difference. The larger fragment will have a difficulty in getting through this small pores, which is actually the material that makes up this gel-like material. On the other hand, the small nucleic acid fragment will have a relatively much easy to move through these small pores, so they will end up faster than the larger fragment. So it is actually highly correlated and to their size, DNA fragment size. The shorter the fragment, they will move faster, and in terms of this the fragment size and the movement relation, the correlation, then you can predict precisely that the pretty much about the size of any DNA fragment. And same thing you can also apply to the case of RNA too. So all these nucleic acid they will end up according to their relative sizes. So in wild type the DNA sample, if you run down your DNA sample after subjecting your DNA sample with to that particular restriction enzyme, and at least as far as that area of the beta hemoglobin the gene concerned, you have 3 fragments. 1, 2, 3. 3 different fragments that you can detect on this so-called gel electrophoresis. Whereas in the mutant, the sickle cell mutation carrying individuals sample, you get have 2 fragments, fragment number 1 and fragment number 2, and actually this fragment number 1 is like the combined version of this the wild type fragment 1 and 2, right? Maybe so for that matter I should actually say this is a fragment number 1 prime, and maybe this is exactly identical fragment number 3 here. So whatever they are, you do have this polymorphism of restriction fragment lengths. Okay, so these reflexes are as we have just seen right over here, the case of the sickle cell mutation. It can be caused by a single nucleotide change, just like only single nucleotide change can generate such polymorphism in cutting. So such single nucleotide change is usually referred as SNP. In these days of DNA sequencing era, a lot of people, a lot of scientists mention this particular terminology of SNP, which simply stands for single nucleotide polymorphism, like polymorphism generated due to the only single nucleotide conversion alteration is what is a SNP. A SNP. Did I pronounce this SNP instead of SNP? Yes. This is another example of just like how scientists usually prefer to pronounce. Instead of SNP, scientists pronounce this as a SNP. Just like the case of RFLP. RFLP and SNPs are the two highly related terminology, in both of which together show some polymorphism among different individuals in terms of their DNA sequence. So here, one such SNP can exist. On this figure is showing that on any given area, for example, like AGT, 3 different nucleotide on the same location of the chromosome or DNA can happen. Or maybe there's another 4th type of SNP, but here this figure is simply showing you an example of what a SNP is. So this is an example of a SNP. So if you try to collect all kind of different SNPs existing in all different humans, it will be just a huge overwhelming number of SNPs can be detected, and some scientists actually trying to identify all possible SNP in terms of any given the gene area or any location of the chromosome of their own interest, and then try to figure out if there is any kind of the SNPs have any correlationship with their respective gene functions is one of the very popular topic item in these days of in so-called genomics study. So there is a so-called some particular branch of the recently appeared scientific branch called the genomics. This is one of such a popular way of studying any DNA sequence variation. Some of DNA sequence variation should have some kind of influence on the gene functions, and without even knowing the gene function, because most of the vast majority of the genes, even after the completion of the Human Genome Project, we do not know what exactly the different genes functions are in particularly. So even without trying to figure out completely each of these genes function, at least if we have some kind of DNA sequence variation on particular gene and you have some correlationship with this gene's function, is there early step of the approach towards the ultimate goal of these scientists. The agenda is actually to know the function completely in every bit of different genes, but we are not there yet. It's just like far away from it. So until we get there, what you can do is at least try to arrange and then figure out some meaningful correlationship is like a current status of so-called the genomic study. Did I go overboard? Or maybe you didn't know actually what's going on over in genomics. So yeah, I will refrain myself from talking too much about this area. So anyway, so as I briefly try to explain how practically we can detect this reflex pattern in different individuals, you have to use the probe that showed you as one of the keywords that we need to carry on in this reflex and the DNA profiling probe and this electrophoresis, gel electrophoresis. But for this to generate this different fragment due to differences in DNA sequence, as I mentioned, we have to use this resection endonuclease. Well, another name for this resection endonuclease is a resection enzyme. This is an enzyme. So basically what the resection endonuclease or resection enzymes are, they can recognize a specific DNA sequence, very specific DNA sequence, but those DNA should exist in double-stranded form of DNA, only DNA. So this resection enzyme cannot cut RNA, which is a single-stranded, and also cannot cut DNA of a single-stranded, but only they can cut this double DNA complementary base paired DNAs and cut it in a specific mode is what they do. And there is some kind of very interesting pattern among those recognition sequences which can be exploited, which are the targets of resection enzyme. They are all in common. They can be said as they exist in a palindromic structure. So that's example of a palindrome. What's palindrome? This is one very good example of a palindrome. palindrome means if you read this from left to right, or if you read backward, then you generate exactly same sentence is what the palindrome means. English version of the palindrome is Madam I'm Adam. If you try to read backwards of this Madam I'm Adam, try it. Madam I'm Adam. Get it? So these are the palindrome. So in DNA world, similar palindrome exists in that this is one such palindrome, GGATCC. And in last week's class, I mentioned that DNA and also RNA, when they have this complementary base pair, they do exist in anti-parallel way, right? So one DNA strand will go from here, from this left side is a 5' end. The 5' means a 5' phosphate group, and this right side is a 3' end. Whereas the partner complementary DNA pair will go, the right side is 5' and the left side is a 3' end. So in this way, then if you read, when you read, usually by convention, you read from 5 to 3' of DNA or RNA. So it's GGATCC, yeah, GGATCC. But if you read the complementary, then same thing, GGATCC. These are this kind of things are examples of palindrome in DNA. So in the case of another DNA sequence, which is a CCC GGG, same thing, CCC GGG. So this is forward and backward identical. Again, it's a palindrome. So all these three different sequence, an example of a palindrome, and there are so many occasions of palindromic sequences on your genome, right? And each of the different palindromic sequences, there are like, I'm not saying, I am not saying that every palindromic sequence on your DNA genome there will be any restriction enzyme that is able to cut. But the fact of the matter is, until now, it's about 3,000 different restriction enzyme have been discovered, all of which were originally discovered from bacteria. And then once you discover any particular restriction enzyme from any different type of bacteria, you isolate the gene that makes such restriction enzyme from bacteria, and then you take advantage, you exploit that gene to make many such protein, and then you utilize. Because why the value of this any restriction enzyme is in that you can use different restriction enzyme in cutting different area of the DNA at your will. So when some time ago, when I was mentioning the plasmid, for example, the plasmid, do you still remember? Speaking of this plasmid, in recombinant DNA technology, you have this plasmid DNA, but the beauty of this plasmid is you can insert let's say this is your gene, human genome, and some area of human genome here, let's say this is you have your insulin gene. What you want is you want to insert your insulin gene only to plasmid like this, okay? So that this plasmid can produce many recombinant plasmid, each of which now carrying the insulin gene like this, and each of plasmid, for that matter, is able to produce insulin protein. In bacteria, then you can produce huge amount of insulin in very cheap prices. So that's the basis of, actually this is the first example of such genetic engineering which happened in about 1975. But the thing is, how do you cut? How can you isolate very precisely only the gene, insulin gene area, and how do you actually implant that gene into any suitable location of plasmid? In both the cases, actually, what it takes is this restriction enzyme. Luckily, you have specific restriction enzyme that can cut either end of this DNA. So it's just a matter of you find a suitable restriction enzyme that targets a very specific DNA sequence area and then cut them out. And same thing, you can also use the same restriction enzyme on this plasmid DNA. So you cut it open and then you paste your isolated DNA fragment together with this plasmid, and that's how you were able to generate this so-called recombinant plasmid. Here, simply I'm trying to explain the usage of the restriction enzyme is so valuable, and since like about 3,000 different types of restriction enzymes have been discovered since then, so practically speaking, you can almost 100% you can cut any DNA fragment in any way if you want, and then do whatever you want in manipulating them. So the point is, restriction enzyme have been very instrumental in the advancement of this genetic engineering, is what I can say about it. And as such an example, this in DNA profiling, when you try to generate such difference in the restriction fragment length difference, now you can also use this restriction enzyme, is the point. So then those fragments generated by appropriate restriction enzyme can be separated in according to different sizes using this mentioned the gel electrophoresis. So this is a technique with which this DNA and RNAs or even the proteins, although the materials using in the protein separation is slightly different from the case of DNA and RNA separation, but basically the same principle of separating them in terms of their different sizes in the electrical field. So this nucleic acid gel electrophoresis generating materials is called the agar rose. It is a type of a carbohydrate abundant in a plant like algae, and the pores. If you have this the mesh of the gel matrix, then you get to generate these small pores seen in this electron micrograph, through which DNA and RNA fragment can migrate, and then obviously smaller fragments can move faster than the larger fragment. Yes. So these gels, the agarose gels, are actually used in various different ways, not only in addition to this making this gel electrophoresis. And this is the photograph, actually how you prepare the material for the gel electrophoresis. So you melt the agarose, and then pour into already set up template, the mold, and then you finally get to have this solid material, and each of this hole you can apply different DNA sample, and after which you apply electrical field where the positively charged is at the end, and the nucleic acid migration starting point is where the negative charge is. So like I said, these nucleic acids are highly negatively charged, so they will migrate towards the positive pole. And smaller size nucleic acid can move faster, like that. So it's just all the way those DNA or RNA fragment ending up at the bottom are the smaller ones, and you have already standardized so that if your DNA sample is somehow migrates some way over here, then you can at least fairly accurately estimate their real size in terms of how many nucleotide base pairs are there in with this type of size is already you have like prepared in some of the standard way. So but still you have one problem. This is usually, let's say, let's get back to the example of this sickle cell, the mutation. In order to identify this reflip between the wild type individual sample and this mutant. How do you distinguish? Like in the wild type, you are supposed to have 3 DNA fragments cut with that particular recession enzyme, whereas this mutant you have 2 fragments. But this is actually the real life example of if you isolate your DNA, and let's say your DNA, which is a 3.3 billion nucleotide base pairs, and then if you cut your entire DNA with one specific particular restriction enzyme, then because your DNA sequence are actually in terms of is almost as good as a randomly arranged, so then the pattern of a DNA fragment cut with any given recession enzyme would be like in every bit of a different size of a DNA fragment will be generated because of the reason. So here, this pattern, all these whitish smear dirty structures are such the representation of each different or random DNA. I shouldn't say it's a random. It is all different size of your DNA is cut with that particular recession enzyme. So usually its DNA fragment cutting pattern is like a smear like this. Then the question is, where is Waldo? How do you identify your actually that 3 DNA fragment, that relevant 3 DNA fragment that represent the area of the beta globin gene fragment? Where are the 3 beta globin DNA fragment? This is a big question. How do you identify them? Well, actually, so this is the original such DNA gel electrophoresis pattern cut with any given particular resection enzyme, whereas this is an A gel, and the DNA sample now cleared, so that now it's only showing the DNA bands or the DNA fragment of interest. How did you remove all this noisy background but only showing this DNA fragments that you want to see? There comes the concept of this probe. So that's why understanding this probe in its concept is extremely important, and also it is a much wider application, but only this narrow area of DNA fingerprint. So we will have to take a look at this and understand the concept of a probe in the second session of the lecture. And now I'm going to finish this first session of the lecture this week.